Hello and welcome back to Biology for Beginners. As always, I'm your host, Morgan. This week we're going to be closing out Chapter 5, which honestly, in my opinion, has been a bit of a weird chapter, and I understand why teachers tend to skip around in books, but the structure of this series, we're just trucking through it and dealing with the weirdness. So we're going to close out Chapter 5 by talking about enzymes. As always, right now, we're flashing up those terms and definition slides for you, so you can either pause the video and grab the necessary vocabulary to understand what we're talking about, or you can pop on over to my Patreon page. The link is in the description down below, and grab my full slideshow as a PDF and have that to follow along, which also includes these terms and definitions. Quick reminder that any level of patron does get the Bio for Beginners a full day in advance, or sometimes slightly less than a full day. You get it in advance, that's the point. So without further ado, let's get into enzymes. I know that I just teased you by saying we're jumping headfirst into enzymes, but first we need to back up for a second and talk about activation energy. So activation energy is essentially how much energy it takes to get those reactants to jump over that hump and move down to the products for the whole chemical reaction to take place. So again, it's the energy that it takes to make the reaction happen. And some reactions are going to require more energy, some reactions will require less energy to happen. So if we're looking at the diagram I have here, the activation energy is that hump there with that bright red line underneath it. That is the energy required to make that reaction happen. So as you can imagine, this is a problem, especially in biological systems. If you're doing a particular lab experiment in, say, a chemistry lab, you can put things on a hot plate or a Bunsen burner to help make that reaction happen because that heat is putting the energy in. But how can a biological system solve this problem of the activation energy hurdle? Well, the solution to this problem is to use an enzyme. And enzymes are basically biological catalysts. Now, if you're working with chemistry, a catalyst is something that kind of speeds up the reaction. But what it's actually doing is it's lowering that activation energy requirement. So this first diagram we're looking at, you'll notice the black line with that very high hump that's represented by the arrow on the right, A, that is the activation energy required for the uncatalyzed reaction. But then if you notice there's that red line below it with the arrows B, that is going to be that much lower activation energy, which is the activation energy with that enzyme or catalyst being utilized. So again, just another little depiction of this that kind of gives you more of an idea of the enzyme kind of structure and function, which we'll get into in the next slide or two. This activation energy is that barrier for those reactants to hop on over to the product level. But then if you add that enzyme in, it kind of picks up those reactants, lowers that activation energy barrier, and allows the products to be produced with less energy input and speeds up that process, because just because there's that high activation energy barrier doesn't necessarily mean the products won't happen. It just means that it's going to take longer, because it's going to take longer for enough energy to be input into that particular reaction to get it to drive forward. So how exactly do enzymes work? Well, first you're going to begin with an empty active site. The active site is basically the part of the enzyme where it all happens. It's usually some sort of like groove or depression in the actual enzyme. And what's going to happen in the next step is the substrate. So the reactants, the things that you start with, are going to bind to that active site. And the bond between the enzyme and the substrate is going to be secured with what is known as an induced fit. So the shape of the enzyme will actually kind of be altered to grab onto that substrate a little more snugly and hold it into place. And then the reaction's going to occur. And then that product at the end will be released from the enzyme, and the enzyme will reset to be used again. An important thing to remember about enzymes is that they are usually going to be proteins, and they are not used up in the reaction. So at the end, the enzyme is still there. The enzyme is wholly unaltered. It goes straight back to the beginning. However, the reactant will be transformed into the products. And on the little diagram here from the book, they're using the enzyme known as sucrase as an example because that is a fairly simple reaction. And enzymes are mostly going to be named with things that end in the ASE. So if you're reading something scientific and a word like sucrase or lactase pops up, you know those are enzymes. And usually they're named to go along with the substrate. So the reaction they're helping to catalyze. So sucrase binds to sucrose. Sucrose is a sugar, and then breaks it down to glucose and fructose through the reaction with water. So again, that's how the reaction works when it is being catalyzed with the enzyme. So enzymes do have limitations. They are a biological thing, and they do require specific temperatures and specific pH. And if you have improper conditions, it'll cause the enzyme to either break down or fail to function properly. Remember that enzymes are usually going to be proteins, and proteins can be very picky about the kind of environment they're in. So if the environment is not correct, it will cause the protein to denature and break down. So the enzymes that are more in your neutral body areas are not going to work if they're all of a sudden in a very acidic environment like in your stomach. But again, the enzymes in your stomach that are particularly suited for that very acidic environment won't work if they're not in the correct pH. And also when you're running a fever and thing, that's why certain reactions don't happen as well, because your temperature is elevated. And the big takeaway is here, there are limitations. Enzymes are picky about their temperature and pH and other conditions around them. As well as having some limitations and pickiness about the pH and temperature, sometimes enzymes need a little help. And they get this help from things called cofactors. Cofactors are going to be these non-protein molecules and ions that just help catalyze the reactions. So even though the enzyme is what's grabbing onto those reactants and acting as the biological catalyst, sometimes you need a little bit more input that kind of helps the reaction happen, even though it's inside that enzyme. And you can have these inorganic cofactors like zinc, copper, and iron, or occasionally you'll get these organic cofactors that are called coenzymes. And again, they're not going to be proteins. They're usually going to be things like folic acid, which we have depicted here. So your vitamins and things that you have to take in your diet, there are some of them that end up as these coenzymes, like folic acid. While it would be lovely for your body to have all these reactions going continuously, it doesn't need that to happen. So you do need some sort of regulation of these enzymes so that the reactions that they catalyze aren't just going continuously and wasting energy and having too many things happen at once. And the way that this regulation happens is through inhibitors. And there are three main types of inhibition. And the first one is going to be a competitive inhibitor. This is something that's going to bind to the active site of that enzyme and not allow the substrate to actually bind and be catalyzed and not allow for that reaction to go forward. So it's in direct competition with the substrate that needs to bind to the enzyme. And competitive inhibitors do mimic the structure of the substrate for that enzyme, because enzymes are picky. Enzymes only bind to the things that they are specifically produced to bind to. So the inhibitor has to mimic that shape of that particular substrate. Another form of inhibition is going to be a non-competitive inhibitor. Now, this is going to be another thing that's going to bind to the enzyme. However, it binds to another site, and then the shape of the enzyme is altered to the point where the substrate can no longer bind. So if we look at the images we have on the right of this slide here, the top image is just a normal function. You get that substrate that is shaped to properly just kind of pop into the enzyme like a little puzzle piece. But the competitive inhibitor mimics at least part of the shape of that substrate and blocks it from binding properly. The substrate can't attach, the reaction cannot happen. And then the non-competitive inhibitor, that little yellow piece kind of pops in another little groove somewhere else on the enzyme, and that shape changes. The substrate no longer fits and cannot access that active site. The last regulation that we're going to talk about today, and the last thing we're going to talk about enzymes, is the feedback inhibitors. This is a product of a specific metabolic pathway. So if we remember from the last talk when we're talking about energy, a metabolic pathway is just a bunch of reactions that happen to get to a specific endpoint. So a bunch of reactions as opposed to just one. Well, this product of the metabolic pathway in this case is going to inhibit one of the enzymes on the pathway. It's not necessarily going to be the one that produced it, and it's not necessarily going to be the first one, but it will inhibit some point of the pathway that will then either stop the pathway entirely. In the case of the diagram here, it inhibits the first enzyme, which stops the pathway, or it will just alter where the pathway goes. When we talk about other processes in later chapters, we'll see this happening where if one enzyme is stopped, the path will then switch to another product being produced. So it doesn't necessarily have to be a competitive inhibitor. It can be a non-competitive inhibitor. It just feedback, a particular product is made, there's enough product that then inhibits another enzyme on the pathway. The pathway is in some way stopped or altered. So again, these inhibitors just kind of help regulate the enzymes. And it's important to remember that the inhibitors don't entirely make the enzyme stop functioning forever. These bonds with the inhibitors are usually pretty weak and they're easy to reverse. And your body and other biological systems do have ways to regulate that as well. So when it's time to start making the product again, the inhibition will be ceased and the reaction will start being able to happen once again. So, little quick recap, enzymes just help make reactions happen in these biological systems like your body. They are regulated by inhibitors, so they can be started and stopped when certain products need to be made so that life can go on as it should and biological systems can keep functioning properly. And they're a little picky about temperature and pH, and occasionally they need help from cofactors. So that's all I really have for you today. We're going to be doing chapter 6 next week, which, if I'm remembering correctly off the top of my head, is cellular respiration. So be excited for that. And I hope you have a wonderful rest of your day, afternoon, evening, night, what have you. And I will see you all in the next one. Thank you again for watching this video. Don't forget to like, comment, subscribe, and ring that bell if you would like to see more. And if you'd like to follow me on any of my other social medias, the links are down in the description below. Don't forget to check out thereptilegoth.com for all of my articles and blog posts. If you found any value in this video and you would like to help support the channel, please check out my Patreon page. That link is also in the description down below. And a special thanks goes out to my Diamond Dragon patron, Diane B. What you're doing is really helping me fund a dream here. I will see you guys all in the next one.